Hello, everybody. I'm here today to encourage anyone who wishes to donate their genetic material to research. All progress since the very earliest days in IVF has really relied upon the availability of human genetic material for research. Animal research just is not the same. Researchers are working on different areas and they will need different things for their research. Now, our group is studying events just before and just after fertilization through to the second cleavage division, which is when the embryo has reached about the 4-cell stage. In my job, I spend a lot of time and energy looking at embryos that belong to people who haven't become pregnant during their IVF treatment. I'm attempting to understand what's happening with those embryos and how we can develop bespoke treatment programs for the people affected. And it's very clear to me that the problems mainly occur right at the start, just as the fertilized egg is trying to become a 2-cell embryo. Now, this stage is difficult to study. Firstly, because eggs are large, they're primed, ready to go through the fertilization process, and they're unstable. So they're at a sort of a difficult stage in their own history. It's difficult to access this sort of material. It's very precious, very special, very rare. And so ideally, what we're trying to access is human eggs at immature and mature stages and very early embryos to enable us to do this research. At the moment, if patients consent, what we're asking for are unfertilized eggs, eggs that didn't fertilize when we tried to fertilize them. So these would be about a day old. And because of that, they're not in prime condition. They might have deteriorated a little bit. So we use them, but they may not be top quality research material. Immature eggs, again, these are eggs that are collected that can't be used in the IVF process. Ideally, they would have been mature at the point when they were collected, so we're not sure that their response is actually going to be normal, but we can still use them and we can still learn from them. And we also use abnormally fertilized eggs, such as triploid or tripronucleate embryos. And these can also provide some useful information, but it's not normal fertilization. Our research has been criticised in the past because we were not using top quality material, so the results may not be reflective of the normal situation. To overcome this, we set up an egg sharing to research programme. Now, this allows patients who are young and with a good chance and probably having plenty of eggs to choose to share some of those eggs with us as researchers. And in return, we will pay for a large part of the treatment that those patients are going through. That payment is from our research grant, so it's all approved through ethical committees and so on. And this would allow us to access fresh, mature eggs, normal eggs from young women, which ought to be top quality material that we can then use in the research. We can fertilize them to make early embryos. And through doing this, we've gained some really exciting results about the first cleavage division that I'm sure you'll be hearing about. And this is helping us now to explore methods that might be able to help eggs through that first cleavage division that might allow us to improve IVF for the future. Now, the patient pregnancy rate from these egg share cycles is good, so we're pleased as well with that we're not disadvantaging anybody who is offering us eggs as part of this programme. And we've shown that even these fresh, mature, normal eggs from young women are still prone to errors and the same sorts of errors that we were seeing in our previous research. So in that way, we've validated a lot of our previous work, which has also been extremely valuable. How can people get involved with this? What can people do to help? We really would value further collaboration and further people being involved in our research. And there are different ways that people could become involved. So first off, there are many people, young women often, going through the process of egg freezing, and their aim is to use those eggs in the future to become pregnant. Now, many young women will in fact find that they can become pregnant without the need to use those frozen eggs. So it's likely that in future, those eggs may no longer be needed by those patients. It would be great if clinics could provide information to those patients about the opportunity to donate those eggs to research if they no longer require them. People who'd like to do this, any patients out there who might be listening, you could encourage your clinics in this, and you could also contact us directly if you wish to. If clinics would like to collaborate with us, please consider whether you might be able to collect any immature eggs from patients who are going through the ICSI process and vitrify those so that they could be shipped to us at a later stage. This would obviously require patient consent, so we could help with that, and any lab consumables that may be involved, again, we can help with that through our research grants. Do let us know if you might be interested, and we can talk about it and find a way that would work for everybody. We're also keen to receive frozen embryos, but for us, because we're working right at the beginning of embryo development, we're really only most interested in those that were frozen at the 2 pronucleate stage. Most embryos nowadays are vitrified as blastocysts, but if anyone out there is still using or still has available those very early embryos that we used to freeze, please consider whether the patients might wish to donate those to research. And finally, we can accept patients onto our Egg Share for Research programme. Now, this is only taking place in Coventry. Any patient who wishes to consider this programme would need a GP referral to Dr Rina Agrawal, who runs our egg sharing programme. She's based at the Coventry Centre for Reproductive Medicine. It's an NHS clinic. So for the future, the best way forward would be for there to be a tissue bank that manages all of the available material arising from IVF so that patients who wish to can donate their material very easily. Researchers who need to use certain types of material can access it in a carefully controlled manner. Banks are already available for all sorts of different types of material, such as stem cells or fetal tissue, cord blood, and so on. But there's none for human eggs and human embryos. And the reason for this is because at the moment, the law doesn't allow us to gain generic consent that would allow, say, some eggs or some embryos to be used in a range of different research projects. Each particular project has to be signed up to by each individual person involved, which involves a lot of work. It's quite a cumbersome system. It would be great to have a centralised bank so that people up and down the country could donate material if they want to use in research. It would streamline the whole process for clinics and for researchers. It would make a much more consistent patient experience of accessing and interacting with research. There'd be more research material available. There'd be fewer hurdles for researchers. Not that there shouldn't be some, but there should obviously not be more that are going to slow up the research and prevent the progress being made. And there should be careful, transparent regulation around the tissue bank. Donating material to research is always voluntary. There's absolutely no obligation. And for people who don't want to do it, that's absolutely fine. All the research that we do in this field is licensed by the HFEA, and it's carefully monitored by the organisations that we work for and recorded through annual reports, for example, to the research ethics committees. This particular project that we're working on now has been running since 2004. And that's 20 years now and counting, so we will be continuing into the future. And over those years, we've had literally thousands of patients who have provided their material to us. And I'd really like to thank each and every one of them for the time and effort and dedication that they've shown to allow us to do the work that we do to hopefully help and make progress. Clinical practice is working right at the limits of scientific knowledge in this field. There are many questions that we don't know the answers to. The only way to make progress, to improve the scientific knowledge about embryos, to improve the success rates of IVF, and to help individuals with their specific fertility problems is through research. And I hope you will support our efforts in this way. Thank you.